Good morning, everyone. I hope you had some time to review the last lecture. So today we will be um, again talking about DNA recombination, and we'll go in more details about site-directed DNA recombination. This lecture is based on chapter 12. The page numbers are noted here. So we were talking about homologous recombination in our last lecture and last class. And I mentioned that homologous recombination generates genetic diversity, and that's quite important for a population and combined because more genetically diverse individuals will be able to tolerate the climatic conditions better, and they will be better adapted because some of these individuals will be better able to survive the climatic conditions, and those will then proliferate. Recombination initiates between identical sequences, and as we talked about in previous set of slides, these are called chi sites. The chi sites are exactly same sequence. These are identical. That's where recombination initiates. But the strands can be similar. They don't have to be identical, and most of the time these aren't identical, and that's where the genetic diversity comes from, because different alleles of the same gene will have a slightly different sequence than the other one, and that's why one is usually dominant and another is recessive. So these sequences need not to be exactly identical, but they need to be very similar. So here's the example. We talked about homologous recombination. Homologous recombination is very common during meiosis because that's where the genetic diversity comes in place. Here in this example, there is this strand exchange, this chromosome arm exchange is going on. So what will happen in this case is now these two individuals will have this chromosome that has the characteristics of both the maternal and paternal chromosome together. So again, bringing this diversity to the population. This homologous recombination is, as I said, it's very common during meiosis, but it also occurs during double-stranded break. So when there's a double-stranded break in the DNA, one of the mechanisms of fixing that break is this homologous recombination, where the cell tries to find some sort of homology with other chromosome and then copies that information to the chromosome that's broken. There are three types of recombination that I mentioned earlier. Homologous recombination that we went in detail in previous set of slides, site-specific recombination, and transposition. This is one of the example of homologous recombination that we talked about. As homologous recombination can involve a single-strand nick or double-stranded break. Most of the time it's a double-stranded break. In meiosis, this double-stranded break is caused on purpose by SPO11 enzyme, which is an endonuclease. In other cases, outside meiosis, this is usually a break. But recombination can occur by both single-stranded nick as well as double-stranded break. So here in this example, this is a single-stranded nick. The single-stranded nick, then it opens by a helicase activity. This will then invade into this other chromosome. And once it's invaded into it's another chromosome, this will copy this information here. It's very similar to each other, so that's why they're showing this homology. This will copy this information, and this information will then be, since it's copied here at some point. So this is the holiday junction here. This holiday junction will then migrate, and this is called branch migration, and this will eventually this will resolve, bringing these two, making again these two chromosomes. And here then you can see that the exchange of information has happened between these two chromosomes. these two chromosomes of the same pair of chromosomes. So it doesn't happen between two very different chromosomes, because different chromosomes will have very different sequences, but the same homologous pair of chromosome will have very similar sequences, because each of those chromosomes, one comes from mother but one comes from father. So those sequence, they have the same genes on those chromosomes, but may have different alleles. Okay. So all of these different different events in the cell, DNA replication, repair, and homologous recombination, they all occur with high fidelity. So DNA replication, as we already saw, that the polymerase has this capacity to check for any errors, and then repair double checks all the errors, and homologous recombination makes sure if there is a double-stranded break to fix that double-stranded break with the homologous chromosome before the cell divides. So it gives us a very high fidelity, which also means, this high fidelity means that the genomes over generations are nearly identical. So there are some changes, but these changes are very few and far in between. And most of these changes are usually beneficial, because lethal changes will be eliminated. Recombination means rearrangement of DNA sequences within this nearly identical genome, and this rearrangement of DNA sequences gives make this genome dynamic, make the genome structure dynamic. So two of these mechanisms that we're going to talk about in today and in next lecture, conservative site-specific recombination and transposition. These are very important for DNA rearrangement, and both of these mechanisms share key mechanistic features. So here, for example, this is an example of site-specific recombination. On top and bottom is an example of transposable element. So in the recombination, this conservative site-specific recombination, there are these recombination sites. These recombination sites that's where the recombinase or the enzyme that will lead to the cut will come and bind. And these recombination sites are important, because without these recombination sites, recombination can't happen. And in this example, the recombination happened, and this just the sequence inverted here. So there's a cut here and cut here, and this now that is attached, or it's ligated in a reverse direction. And with what that means is now these sequences, the genes are switched. If we look at transposable elements, as you know, these are called jumping genes. These jumping genes can jump from one place in the genome to another place in the genome. These jumping genes can act two different ways. They can insert themselves in a different position in the genome by cutting from one. So they can be cut and paste, or they can be copy and paste. So by both ways, they change the genome structure. So here in this example, the transposable element deleted itself from this site and inserted in a different site. So now this sequence is different from what it was earlier, and similarly this sequence is different what it was earlier. So these can bring this more dynamic genome structure into a very identical genome. So genome is still the same, it's just the position of these sequences has changed. Site-specific recombination requires these recombination sites, as I mentioned. These recombination sites are specific short sequences where DNA exchange occurs. These recombination sites consist of a recombinase recognition sequence. This recombinase recognition sequence is usually short, around 13 to 20 base pair, and that's where recombinase binds. And these recombination sites so these are present on both sides. They flank a shorter crossover region. This crossover region can be 4 to 8 base pair, sometimes a little longer, but usually smaller than the recombinase recognition sequences. So so there are two of these recognition sequences flanked by this small crossover region. So here in this example, these are recombination sites. These recombination sites are where this is not a very good example. I will show you next example in a next slide what these recombinase recognition sequences and crossover region look like. The two families of recombinases, serine recombinases and tyrosine recombinases, that have been identified and studied so far. The mechanism of these recombinases is quite similar to TOPO2 or SPO11. Remember SPO11, that causes these double-stranded breaks in meiosis. These enzymes also hydrolyze for a diester bond. They act as endonuclease and then reform it or ligase. So there's no real net energy transfer occurs because this uses the energy of hydrolysis of the bond to reform the bond. Depending on the class of enzymes, these cut can be a double-stranded or single-stranded. However, the single-stranded is not quite exact term. It's a single-stranded break at a time, although both the strands will be broken eventually. Serine recombinases cause a double-stranded break, so it just breaks both strands at the same time, whereas tyrosine recombinase breaks one strand at a time, but it will break both strands eventually. One of the examples of the site-specific recombination is integration of bacteriophage lambda genome into the bacterial chromosome. So here, this is the phage recombination site. Phage, as you remember, bacteriophages are viruses. These viruses infect bacteria. So in this example, this bacteria is E. coli, is attacked by invaded by this bacteriophage lambda. This lambda genome comes inside the bacterial genome, but in order for so the viral genome goes into two different cycles, right? A lysogeny and lytic phase. In lysogenic phase, it stays within the bacterial, embedded in the bacterial chromosome. So that means it has to first recombine, it has to first integrate itself into bacterial genome. In lytic cycle, when the virus particles are rapidly multiplying, and then they will be making the protein coat for themselves, and so they will break the cell and come out. At that time, they have to come out from the bacterial genome. So this integration and excision from the bacterial genome can be done by the site-specific recombination. So here in this example, this is the phage recombination site. This is the bacterial recombination site. These recombination sites will allow this phage to recombine with bacterial genome. And here, this is the bacterial genome. The phage genome is now inserted within the bacterial genome. So site-specific recombination can generate three different types of DNA rearrangements. Insertion of a segment of DNA that I just showed you, for example, this bacteriophage DNA integration. Deletion of DNA segment, you can again think of the lytic cycle of bacteriophage DNA, where this DNA segment, the viral DNA segment, has to be deleted from the bacterial genome. And inversion of DNA segment, that means instead of any addition or deletion, the sequence just inverts, and that way it rearranges itself. Example that we saw earlier. Each of these recombination sites are organized as a pair of recombinase recognition sequence, as I mentioned earlier. Could be 13 to 20 base pair sequence long, and there's a central short sequence that these recombination recognition sequence flank, called crossover region, which is usually smaller, around 4 to 8 base pair. So here, this is a good example of these recombination sites and crossover site. These two are the recombination sites, and this is the crossover site. So these recombination sites, you can see. So this recombination site, this is the crossover. That's where the crossover, that's where the recombination occurs. your recombinases, the enzymes will bind to these both sides of this flanking region. Recombinase recognition sequence is here. This and this here, and this is the crossover region. In this example, this is a 4-base pair crossover region. So recombinase binds to recombinase recognition sequence. So this makes an dimer. Together it makes a tetramer. Recombination happens, and here this recombinases will make a break in the DNA depending on whether it's a serine recombinase or a tyrosine recombinase. It will make either a double-stranded break or one-strand break at a time, and then this strand will exchange. They will recombine, and ligase will again fix the sugar phosphate bond, and here you can see that these have been recombined. This portion of this is from this chromosome, and the other portion is from other chromosome. So again, as I mentioned, there are 3 different kinds of DNA arrangement that are carried out by these site-specific recombinations. Insertion of a segment, deletion of a segment, and inversion of a segment. This is an example. Again, let's consider this as a viral DNA particle. This is a bacterial DNA. Only a portion of bacterial genome is shown. And these are the sites, these crossover sites. They have similar sequence. These sites will find each other. Once they find each other, once these find these align to each other. Once these align, the crossover will happen at this crossover site, and this crossover will allow the sequence to be inserted into the bacterial genome. So here the viral genome is inserted within this bacterial genome. Now let's consider the virus goes into lytic phase. Then this genome need to come out in order to be packed into the viral capsids, and here then this again these crossover sites will align themselves. The crossing over will happen, or the break will happen here at this point, and these two will combine with each other, and these 2 sites will combine with each other, and these 2 sequences will now be separated. It's deleted from that sequence. This is an example of inversion. Here, this sequence is just one sequence in a genome. This is not. Let's not consider it as a viral or bacterial genome. It's just the same genome. It has these 2 different genes here. Say, for example, C and D. These different sequences. This is the crossover sites here. These crossover sites will the recombination sites on each end, here in orange, here in blue. These recombination sites will then recombine. These will find this portion where the crossover will happen, and these switch this sequence instead of deleting or inserting anything. So now instead of it's going in this direction, this is going in a reverse direction. So this sequence has been inverted. So all of these different ways will change the DNA, not the genome sequence per se, not the genome genome information, but where these different genes are in the genome that will change. Okay. Tyrosine recombinase mediated recombinations are best understood, so that's what we will be talking about today, are the tyrosine recombinase mediated recombinations. One of the best example of tyrosine recombinase mediated recombination is Krey-Lox recombination. Most of you probably already heard about Krey-Lox systems, because the Krey-Lox system are being used in a lot of different labs in order to carry out these site-specific recombinations and in order to study gene functions as well. The Krey-Lox system consists of 2 components. This system is derived from a P1 bacteriophage. So again, it's derived from a virus. It consists of 2 systems, 2 components, a Krey recombinase and LOXP recognition sites. So these are specific recombinases. These are called Cre recombinases. Okay. P1 bacteriophage uses these components as part of its natural viral life cycles, but the researchers have adapted these components for genome manipulations in order to understand gene functions and also to carry out specific mutations that they want to carry out. So Cre recombinase, which the Cre comes from, causes recombination. It's a 38 kilodalton protein which can carry out intra and intermolecular recombination at the LOXP recognition site. What does that mean is that intra and intermolecular recombination means it can be within the DNA, same DNA molecule, or between two DNA molecules. So it can do both. The Cre recombinase, it acts independently of any other accessory protein, so it does not require anything else. All it requires is Cre and LOX. And because it's such a simple mechanism, it doesn't require any other cofactors, it doesn't require anything else. It has broad applications, a variety of experiments it has been used. This is really hard to see here, but I will come back to this figure. This shows here. These are the Cre recombinases. These Cre recombinases are bound. This is the LOX site, because that's where. So again, these are the recombinase recognition sites, recombination sequences, and this is that core sequence that we were talking about. That's where the crossover happens. So this is what the Cre-LOX system is. The whole system itself is LOX. This LOX has these two sides on each side for the Cre to bind, and this Cre will cut the LOX in the center, in the middle. Since it's a tyrosine recombinase, it breaks one strand at a time, not both strands at the same time. So this will break one strand at a time. So it's cut this strand here, and this one cut this strand here. So what will happen now? There is a free hydroxyl group because of this break here, because this break happened here, and these will combine. These both ends will combine. So this makes this tetramer of these Cre recombinases. So these are two Cre recombinases on each side of one DNA molecule. So total 4 tetramer. Then there will be second cleavage happening. So this tyrosine molecule will make a break here. This will make a break here. Now this will then bind to each other. So here, now these are separated, and these are recombined. LOXP, which is locus of X or crossover, that's where the LOXP name comes from. So in P1 bacteriophage, these recombination sites are 34 base pair long sequences. This consists of 13 base pair of palindromic repeats. So these are your binding site for recombinases. So this is the 13 base pair long palindromic sequence on each side, and this is separated by this 8 base pair long asymmetric core spacer sequence. So this is, as I earlier mentioned, it can be 4 to 8 base pair, and so these are the recombination sequences. That's where the recombinase will bind. Because this LOXP sequence does not occur in any known genomes other than these P1 phages, so this is a really so if we add this in an individual, in an organism, that means this is really going to be a foreign DNA that we don't have. So there is not going to be any competition between, or there won't be any recombinases binding to these sequences. So here, because, and this is a long sequence, it's a 34 base pair long sequence, because it's so long that there is it's not possible for randomly occurring in any genome. So these LOXP sites can be inserted in any organism. And that's of course the organism has to be amenable to transgenic formation. But if you can add it into the embryo, if you can transform an organism, you can add these LOXP sites in that organism. So here again, this is the same figure that was very tiny in previous slide, but I broke it into different parts. So this is the LOX site. This is LOX B. LOX B site has these Cre recombinase binding sites and this core region. This is the LOX region. These tyrosine recombinases, these Cre recombinases, they come and bind. They make one strand break at a time. So this and this are breaking one break here, and these then bind to each other, these free ends. And here, then this other recombinase will make a break. This is a second strand break. And the second strand break will then again will be bound to these free ends, and resulting into these recombined segments of DNA. The Cre recombinase is catalyzed the site-specific recombination event between two LOX B sites. So this has to be, these LOX B sites need to be present. These both 13 base pair repeat sequences on a single LOX B sites are recognized and bound by the Cre proteins. They form a dimer. Then these two LOX B sites, they align in parallel orientation, forming 4 Cre protein to form a tetramer. And here, this is what it means. So these here and here on both sides, this is a LOX B site, this is a LOX B site. These will they will find each other, these sites, and then these will bind as a dimer here and dead dimer here, so the total a tetramer. So it makes this tetramer. That means 4 Cre together. Double-stranded DNA break, which is not exactly a double-stranded DNA break, but single-stranded and then single-stranded again. So both DNA strands will be broken, but one at a time. These occur within the core spacer of each LOX B site, and the 2 strands will be ligated, resulting in a reciprocal crossover event. So here is an example of how these this is used in outside the viral genome. So this is a mouse where the Cre gene, the gene that makes these Cre recombinases, these enzymes, has been inserted. So this mouse doesn't have the gene that makes the whole LOX sequence, though it only has the recombinase. So by itself, even if it makes recombinase, there is no LOX site to bind those recombinases to bind to. This mouse, this is LOX B, which is called usually floxed. Mouse has LOX B gene, LOX B sites added at about 2 sites. So instead of 2 different genes, it's on the same gene. And this is the target gene that the whatever this gene is that the researchers want to manipulate. So here you will see that this is the promoter sequence. So the promoter is always shown this way. This is the first, that's where the transcription will start. And there is a stop sign, not stop sign, there's a stop codon here. So what will happen is the promoter will initiate this gene transcription of this, then it will stop here, and then there is this EGFP, enhanced green fluorescent protein gene. This gene will not be made if this is not functional, because the stop is right here. The transcription will stop right here. This EGFPs will not be formed. However, if you remove this LOX from this LOX, then there is no stop, and the transcription of this enhanced green fluorescent protein will happen, because the next stop codon is after this enhanced green fluorescent protein. So now let's see, by itself this mouse is just a normal mouse. By itself this mouse is just a normal mouse, because they don't have the complete system. This mouse has the capability to make the enzyme, the Cre and recombinases. This enzyme has capability of binding that Cre enzyme to LOX B sites, but no enzyme, no gene that makes the enzyme itself. But if you cross these 2 mouse, there will be their progeny. Some of these their progeny, some of the litter mouse will have both Cre and LOX. Some will not. Some will be either just Cre or just LOX, but some will have both. So here in this example, this is a Cre LOX P mouse. So what happens in this is, in F1 generation, if this LOX B, if the Cre if this is a Cre-lox mouse, then this Cre recombinase will come and bind to these LOX sites. This once it will bind, it will make these sites unavailable. Now it will recombine. So here this Cre, so it combines the LOXP, take out this target gene. Now this promoter will be able to make this EGFP. This enhanced green fluorescence will be transcribed now, and you will be able to see green fluorescence in these mice. That will tell that because this reporter gene is showing up, that means that your animal is transgenic. If there is no transgene activity, if this mouse doesn't have Cre, just a LOX, then it will not be able to bind to these LOXP site. It will not remove this portion, or this will not make this will not disrupt the function of this gene. It will not make the EGFP because there is a stop codon here. So original gene function is untouched. There is no green fluorescent protein showing up in these mice, and you would know that these mice are not transgenic. So it's easier by using these fluorescent markers. These reporter genes are usually what these are called. This because they report to you whether it is transgenic or not. It's easy to screen a lot of animals easily and know whether these are transgenic or not. Okay, so let me play this video here on Cre-lox, if I can, here.
The Cre-lox-P system is a technique used in a variety of research studies that allows for spatial temporal control over gene deletion, inversion, and translocation. The technique utilizes the Cre recombinase enzyme to catalyze site-specific recombinations. Due to its efficiency in manipulating genes and chromosomes in a wide range of organisms, it is a powerful tool that is widely used in modern-day biochemical and molecular biological research. Previous genetic knockout experiments were restricted if the genes of interest were necessary for embryonic survival, or if the gene in question functioned in multiple tissues or cell types. Cre-lox made it possible to conditionally knock out genes solely in subsets of cells where Cre recombinase is expressed. Cre recombinase is named because it creates recombination. It is an enzyme derived from the P1 bacteriophage and is designed to cut out genetic sequences between two LOXP sites. The Cre recombinase enzyme contains an active site consisting of a catalytic triad with the residues arginine 173, histidine 289, and arginine 292, and nucleophilic residues tyrosine 324 and tryptophan 315. This allows for Cre recombinase to bind to its substrate DNA. The name LOXP comes from the locus of crossover in P1 bacteriophage. LOXP is a site on the P1 bacteriophage consisting of 34 base pairs. This includes two symmetric 13 base pair sequences and an asymmetric 8 base pair sequence. Because of the asymmetry of the LOXP site, Cre recombinase can either catalyze a deletion, inversion, or translocation of the genetic sequence between the sites, depending on the orientation of these LOXP sites. LOXP sites going in the same direction results in deletions. LOXP sites going in opposite directions can result in inversions. Interchromosomal recombination can also result in translocations. There are two main ways to get Cre recombinase to be expressed in a cell. The first is to use a virus to introduce the gene for Cre recombinase into the cell. The other method is to generate transgenic mice that insert the gene for Cre recombinase after a specific promoter. Because this promoter can be expressed in certain cell types, the expression of Cre recombinase can also be limited to these cells. There are many applications of Cre, one of which is lineage analysis, which is the determination of the origin or fate of cells. One example of lineage analysis was used by the Melton lab in determining the origin of adult pancreatic beta cells. They used an inducible version of Cre to determine whether new beta cells were derived from stem cells or pre-existing beta cells. In their experiment, transgenic mice were generated in which the insulin promoter drove the expression of tamoxifen-dependent Cre recombinase. the enzyme that existed in the cytosol as a complex with the estrogen receptor, and heat shock protein. When tamoxifen was injected in the cell, the Cre-ER complex separated, and the Cre recombinase entered the nucleus to perform recombination. Once in the nucleus, the Cre recombinase cut out the LAC-Z stop sequence, and thus allowed the expression of the HPEP label, short for human placenta alkaline phosphatase. Through this process, cells that normally produce insulin, namely the pancreatic beta cells, were tagged with the HPEP label. Using these mice, the Melton lab performed a pulse-chase experiment, the pulse being the set of cells labeled by HPEP using Cre recombinase in the pancreas, and the chase being the set of cells that had the label after a given amount of time. Starting with a population of fully labeled beta cells, they hypothesized that if new beta cells arose from pre-existing ones, the percentage of stained cells wouldn't change. However, if they develop from unlabeled stem cells, the new population will have a much smaller percentage of labeled cells. Their data show that the percentage of labeled cells remained constant, which suggests that new beta cells were derived from pre-existing ones rather than from stem cells. Thus, the Cre-Lox system can be used for numerous types of modifications. Because it can be designed in many different ways, it has been used in a variety of different research studies that involve many different tissue types and organ systems. Perhaps in the future, this technology can be used for cures and treatments in humans. There is
Alright, so that was about Cre-Lox system, how people have used Cre-Lox system in understanding spatial and temporal expression of different genes and different conditions and answering different questions. But then you might ask a question, why when we were talking about CRISPR, why CRISPR is so powerful when there is so many other ways to make transgenic, to study gene functions. But you might have noticed here, in order for Cre-Lox to work, you should be able to make transgenic animals. But that's the biggest step, biggest roadblock for most people who are working on non-model organisms, as it's really hard to make transgenic organisms and make stable germline transformation. CRISPR does not require that, so it's much easier in that way. Another thing with Cre-Lox system, you might have noticed that in order for Cre and Lox to be expressed in different tissue types, it need to have a well-described, well-known promoter that you will tag with it. A lot of non-model organisms don't have endogenous promoters known for them, and these endogenous promoters, without the knowledge of endogenous promoter, it's really hard to know which promoter to use for different tissue types, different cell types, or even for embryonic development. So these are a couple of roadblocks that need to be that made the Cre-Lox system a popular tool for model organism or organisms that we already have tools for transgenic development. For example, mice, Drosophila, Rhabdopsis, all these systems already have tools to make transgenic, and that's where Cre-Lox is pretty beneficial, because now you can make these site-directed changes instead of making a classical transgenesis where you depend on transposable elements, and because they jump from one place to another place, you have to make a lot of transgenic in order to find one that is what you wanted, what you needed. Okay. Another example of site-specific recombination is lysogenic lipogenic cycle in lambda phage. So bacteriophage lambda, which, and again, it's a virus. It's a virus that infects bacteria. It infects bacteria, and it results in either lysogenic phase, which is a quiescent phase, or lytic phase, which is a phase manipulation. That means the phage is ready to come out of the cell. Establishment of lysogen requires the integration of the phage in DNA into host chromosomes, and that's where we started it from in this chapter. When the phage leaves the lysogenic state, or the DNA need to be excised from the host chromosome. Integrase or lambda integrase or lambda int, that's a tyrosine recombinase which catalyzes the recombination between these attachment sites at ATT sites. There are two set of attachment sites, one attachment sites in phage, the ATTP, and the attachment sites in bacteria are called ATTB. So this is a little different here in lambda phage compared to what we talked about previously with these P bacteriophages, where they use Cre system. Cre system is just requires Cre and LOX, so it's much simpler. It does not require any other accessory proteins. Whereas this lambda integrase requires accessory proteins. For example, it will require an integration host factor or IHF to generate ATTL and ATTR of the lysogen. So it's a little more complicated to use as a tool in molecular biology, although it's still being used in model organisms, but not as common as Cre-LOX system, not as simple or straightforward as Cre-LOX system. So here is the example. This is, again, this is your phage DNA, and this peachy color, this blue color is your bacterial DNA. These are ATTB sites. These are recombination sites in the bacteria. These are the recombination sites in the phage DNA. So here, this is the crossover site. So this crossover site will align with each other. These are the flanking recombinase binding sites. These recombinase binding site that's where the recombinase will come and bind. This is the recombinase is integrase here. This integrase will come and bind, but this integrase is not just the integrase is required, but it also required these integration host factor. This integration host factor will help with this integration as well. Then Axis is another protein complex that is coded by transcribed by the plasmid, by the phage mid, by this phage here, and the function of Axis is to delete it, to excise it when it needs to be excised. So during lytic phase, when it's need to excise, then it will be the Axis will carry out that function. So here, let's just go in a little more detail here. This is the circular chromosome of bacteriophage lambda. This is the bacterial chromosome. Bacterial chromosome is also circular, but it's only a part of bacterial chromosome has been shown here. These are the attachment site sequences. So these are those the crossover sequences, and along with the recombinase sites. So these sequences will find each other, and the integrase will bind. So this lambda integrase will bind. This makes this lambda integrase protein complex. So this will bring both of these together, the phage DNA as well as the bacterial DNA. Once these are together here with these integrases, because these are endonuclease, these will make a double-stranded cut. And then they will join these two portion of these. This will make a cut here, which combine with this bacterial DNA here. Integrase will dissociate after that, and this heteroduplex will stay up there. Now bacteriophage DNA has been integrated into the bacterial chromosome. This process can be reversed to initiate lysis, and that will be XS, which is coded on lambda, as I showed you in the previous slide. These phage integrases are quite important. These are great tools as well, even though these are a little more complex than Cre-LOX system, but they're still being used quite a bit in molecular biology. These tyrosine integrases, as I said, such as this lambda integrase, these are quite versatile tool for DNA cloning as well as assembly in vitro and in vivo. Serine integrases, which are not very well understood, they are there are also very few that have been studied. These are efficient in recombination and heterologous hosts, so different hosts, and because they use very short recombination substrate, they're directional and they do not require host factor, just like Cree. These are these are used wherever they have studied. They have been used as a tool. So this is a journal, Plant Physiology, and it has these the image of the cover page was recombinational cloning with plant gateway vector. And when something makes up to the cover page of a journal, that means it's pretty important. So that so you can see that these tools are quite important in order to generate recombinant DNA for cloning of DNA. Mating type switching in yeast is another example of site-specific recombination. Although this event neither adds any new genetic information nor it deletes any genetic information, it doesn't involve any holiday junction, but what it does, it results in gene conversion. And we have talked about this quite a bit in the class already. Saccharomyces cerevisiae has two haploid types, two sexes you can call them, A and alpha. These A and alpha genes are controlled. They control two phenotypes. They compete for the same locus, MAT, to be expressed. So the haploid individuals will be either A or alpha, but if it's a diploid, it needs to have A and alpha both. Two A's or two alphas cannot make a diploid organism. So when conditions are changing and so the diploid organism need to be made, diploid yeast, then if there are all A's or all alpha, some of those yeast cells have to switch mating in order to make a diploid organism. Because copies of both of these genes exist on the chromosome already, for alpha as HML and or A as HMR, and these act like reservoirs. But both of these sequences do not express, only one express, depending on which one the MAT has in it. So MAT always contains a copy of either A or alpha, or whatever the MAT shows is the phenotype of that yeast cell. So here this is HML, which codes for alpha allele. This is HMR or type, or this is A type. Both of these are silent, and whatever is there in the active in MAT site will be the active. So here this is A mating type because A is in the MAT. But if that needs to be switched, and then it can recombine with the sequence and so copy the alpha sequence, and it will turn into alpha mating type. These can switch back and forth between A and alpha. So here again, this is MAT. These are both are silent cassets of HML or HMR, alpha or A. Alpha can go switch the A from MAT and become alpha type. This when there is an alpha, A can go there and may change it into A type. So these can switch. A to A or alpha to alpha will be very, very rare because that doesn't make any sense. It will not change anything. So how it works is we've already talked about this. This requires an HO endonuclease. This HO endonuclease is site specific for MAT. It only works at MAT. So this is specific endonuclease for MAT. It will make a double-stranded break in MAT. Once it makes a double-stranded break, it will work like restriction endonuclease, but it's specific for MAT. Once this makes this double-stranded break, then the 5' ends of that strand will be cleaved to make a 3' overhang using MRX. Then there will be strand invasion with red 51 and red 52 proteins. This is very similar to what we already talked about, homologous recombination, so it will be a homologous recombination here. The strand invasion occurs either at HML or HMR locus, depending on which one was in MAT and now what it's going to switch to. And since this MAT locus now consists of single-stranded DNA, because 5' processing has been happened, and they removed the single-stranded DNA of what it already had. It will copy the gene that it will going to be combined with. So here is an example. Here this is a mat that is showing that is expressing A right now. This A need to switch to alpha. Alpha is right here, this alpha and A, these, because these are alleles of this is a very, very similar sequence, not exactly same, but very similar, because they're going to be the mating type. So these A and alpha, they find each other, they combine these sequences. Each endonuclease will make a cut, double-stranded cut, and will cleave the sequence. And the exonuclease will then cleave the sequence. And this will, because it's recombined here, it will copy the information from here, from this cassette, and then this will result into, because it copies the information from alpha, it will be, mat will be showing alpha type here. Okay. So this was about recombination, site-specific recombination. The next type of recombination is transposition, or transposable. How transposable elements can change the different, can modify the genome at different portion, at different sequences. This is a specific form of genetic recombination that moves genetic element from one site in DNA to another. And you probably know her, Dr. Barbara McIntosh. She was the one who identified, who discovered this phenomena, these transposable elements. She worked on maize or corn, because corn has a lot of transposable activity. Here you can see that all these different kernels, the colors are because of transposable elements. The transposable elements moving from one place to other place in the genome and giving these different kernel colors. These are also called mobile genetic elements or jumping genes. Transposable elements or transposons. So we will cover these transposable elements or transposition recombination in our next class. Okay, so for now this is the last slide, and I hope you all are staying safe and doing well. Whenever you have time, please send me an email or a note on Web Campus that you all are doing well.